Boa tarde a todos, bom dia a todos, perdão. Sejam bem-vindos a mais uma live do Programa de Pós-Graduação. A gente vai começando. Eu gostaria de verificar se tem alguém online para poder verificar se a gente está conseguindo fazer a transmissão. Estamos hoje com a professora Mathilde GENDRIN, do Institut Pasteur de Guyane. Eu vou apresentá-la logo em breve. Gostaria que alguém desse algum retorno no chat para verificar se a gente está fazendo a transmissão apropriadamente aqui, tá? Se estão conseguindo nos ouvir e nos ver. Okay. Can you hear me, Mathilde?
Yes.
Okay, good morning to everybody and welcome to one more live BAIP promoted by the Programa de Pós-Graduação em Biologia de Agentes Infecciosos e Parasitários from the Universidade Federal do Pará. We are transmitting from Belém, Brazil at 11 o'clock exactly via the PPG BAIP YouTube channel. Thus, this transmission can be accessed from anywhere in the world. We also invite the audience to share this meeting. There is still time to do that to other scientists and students that you presume that are interested in these subjects. The highlights of this present presentation of Mathilde concern to the tripartite interaction between the mosquito and its microbiota. And I reinforce that although the knowledge on the mosquito microbiota is continuous expanding, several aspects have not been completely elucidated and yet represent the current challenges in this field. So a brief presentation of Professor Mathilde GENDRIN. Elle a réalisé sa thèse sur l'immunité des drosophiles au CNRS et à PFL. Elles ont ensuite formé la biologie des vecteurs lors d'un post-doctorat au sein du laboratoire Vigilab à Imperial College London. Elle a rejoint l'Institut Pasteur de la Guyane en 2017 pour monter le groupe à 5 ans qui s'appelle Microbiota of Insect Vectors afin d'étudier l'influence des microbiotes des moustiques sur la capacité des moustiques et transmettre des agents pathogènes. Elle a aussi obtenu sa HDAC dans l'Université de Paris en 2019. Dr. GENDRIN prepared about 40 minutes of presentation, and after that, we will be open for further discussion with the audience. Questions and comments can be written during the presentation directly into the chat of YouTube channel. It's also une opportunité pour dérouiller mon français. Pardon. So, Professor Mathilde, you can present now, and you have your time to present your expertise about the curious title, toolbox. Very interesting. So, you can start again. You can start now, sorry.
Thank you, Tiago. And thank you for giving me the opportunity to present our research. As Tiago just said, I'm leading a group at Institut Pasteur de la Guyane and I've started around 4 years ago. And when I look at it right now, I think that most of our questions on the mosquito microbiota are still unaddressed, but it looks like in the last 4 years, we've been able to prepare tools to be able to address our questions. And this is what I will speak to you about. So to introduce first who we are a research group called the Microbiota of Insect Vectors group, present in the Institut Pasteur de la Guyane. You can see on the right-hand side where Guyane is. It's in French Guiana, a region of France bordering Brazil and Suriname. And we are in this Institut Pasteur de la Guyane, part of one of these Pasteur network constituted of 33 institutes all around the world, and we are here in the only one in the Amazon forest. And the good thing about being here with this location is that we have advantages of being part of France and Europe, where we had the chance to build really great infrastructures to work in the lab, and also being part of South America and on this hotspot in French Guiana, where we have around 235 different species of mosquitoes, and which allows us to do field work in really excellent conditions, where sometimes doing field work is just going on the campus to collect mosquitoes, so in a very simple way. And sometimes, as you can see here, we even have to build our lab in a different place to be able to collect our mosquitoes and sample them. Why do we study the mosquito microbiota? We study mosquitoes because, mostly because they are disease vectors. We know that dengue, which is transmitted by notably Aedes aegypti mosquitoes, is responsible for 400 million cases a year, and malaria for 200 million cases a year. Malaria is transmitted by Anopheles mosquitoes. And so there is this causes a real burden for human health and also for the economic development of the countries which are under the pressure of these diseases. Of course, it's only two examples of diseases, and many other vector-borne diseases are present in these regions in tropical countries. Mosquitoes, of course, cause a nuisance for human populations as well. And we focus mostly on the mosquito microbiota because it has been found to have a huge impact on the mosquito life cycle. I will speak a bit more deeply on it a bit later. It's notably very important for larval development. And it has also some strong impact on transmitted pathogens. It has been found that the microbiota naturally protects mosquitoes against Plasmodium infection and thus against potentially malaria transmission. And also some bacteria of the microbiota may have positive or negative impact on virus infection in the mosquitoes and have some impact on these arthropod-borne viruses. And also another reason why it's interesting to work on the mosquito microbiota is because the microbiota is a source of variability between labs. So even if you don't care about the microbiota, sometimes having some tools to standardize the microbiota problem can be interesting. And also there are variability between lab and field conditions, and it's also interesting to look at it and to consider having some relevant models to work on mosquito and mosquito-borne diseases. As I told you, the mosquito microbiota has a high impact on its host and on the host physiology, and this both at different stages during life cycle. In larvae, it has been found to be very important for larval development and participate in the nutrition of larvae, and in adults to participate in the nutrition, immunity, vector competence, lifespan, and reproduction, and having different impacts, positive or negative, on all these traits. And it has also been found that actually the impacts which are found in adults may also be due to carryover effects. So what we call carryover effects, it's effects which are linked to the microbiota which was present in larvae. And it has been found, for instance, that if you expose larvae to different microbial environments, then when they are adults, even if they share similar, during adulthood, similar microbiota, they will have carryover effects, which are due to the microbial environment during larvae. So the size has been can be affected, this immunity and vector competence notably. And if you want to study the host microbe interactions, there are two ways to look at it. One is to look at presence, absence of the microbiota, and the other one is to focus on the composition. If we look at the presence, absence, you may want to do antibiotic treatment on your host, which will allow you to get rid of most of your microbes, and then you can kind of do some presence absence, but it's not really perfect. And the other way is to produce germ-free hosts, and in our case, then germ-free mosquitoes. On the other side, when you look at composition, you may be interested to look at the impact of environmental parameters, such as the mosquito origin or mosquito species, whether mosquitoes are genetically modified, or many other parameters which may affect the environment of the mosquito microbiota and thus its composition. And then these parameters may also, via affecting the microbiota, correlate with different life history traits, such as the lifespan of the mosquito or its vector competence. And another thing that we can do when dealing with composition is to produce notobiotic hosts. An autobiotic organism is an organism with a known microbiota. So to produce them, in general, you produce germ-free hosts, and then you colonize them with microbe of interest. And so then, by varying the composition of the microbiota in your notobiotic mosquitoes, you may be able to analyze the importance of microbiota composition on different traits. And I will introduce several tools that we have set either on these aspects or on different aspects throughout my presentation. The first one is the hammer. It's the production of germ-free mosquitoes, and it has been developed by Ottavia Romoli, who used to be a postdoc in the laboratory until recently. And this has recently been published earlier this year in Nature Communications. The problem we had to deal with the production of germ-free mosquitoes is that if you sterilize eggs, you can do that easily using bleach and ethanol, different washes. Then you will be able to obtain some germ-free larvae, which don't carry any microbiota, and they will be perfectly happy, but just the problem is that they will not develop further. They can stay alive for several weeks, just will not develop. And it has been shown that by using several types of bacteria, for instance, Escherichia coli, you can rescue development, and then you will get some development within similar timing as what you have normally with conventional reared mosquitoes. And the hypothesis we decided to test is that we could use transient colonization to produce germ-free mosquitoes. So transient colonization is a system based on the use of Escherichia coli, which is oxotrophic for two amino acids, meso-deaminopimelic acid and D-alanine. And these two amino acids are really essential for the building of the bacterial cell wall. So as long as you provide these amino acids to this strain of Escherichia coli, it is growing perfectly well, but as soon as you remove it from their diet, they will not be able to grow anymore. And so if they are in a host, they will be maintained. Our hypothesis with the mosquitoes is that they would be maintained as long as we give this supplementation, and as soon as we remove the supplementation, we would get some germ-free mosquitoes. So We tested this by adding some Escherichia coli, either the wild-type version or oxotrophic version, and comparing the success of development of these larvae compared to conventionally reared mosquitoes that you can see here on the left-hand side in black. And we saw that actually whether we had germ-free, whether we recolonized our germ-free larvae with wild-type or oxotrophic bacteria, we would have a very similar development success, which was really high and even slightly higher than what we observed with conventionally reared mosquitoes. Then, are these mosquitoes indeed germ-free? We tested this by doing colony-forming units assay in just after emergence of our mosquitoes. You can see here on day 1, day 2, and day 3 after emergence. We saw that all the mosquitoes which did carry the oxotrophic strain of bacteria turned sterile, as we did not detect any bacteria on after emergence. Then and if we compare that to those carrying wild-type bacteria, half of them still had 10 to 1,000 bacteria per larva per adult after emergence. And then we went back to know a bit what are the dynamics of colonization in larvae and pupae. So during the immature stages, there are 4 different larval instar in mosquitoes, and we started with the 3rd larval instar. We saw that at this stage there was no significant difference in the number of bacteria present in each type of larvae. All of them were colonized, and the number started to decrease at the end of development in both cases, and this is known already that during metamorphosis here, during the stage of pupation, there is less bacteria than what you would have in larvae. And in the case of pupae, there is in the case of oxotrophic bacteria carrying mosquitoes, we would see that at the 4th instar, and then even more strongly during pupation, we had a really very small number of bacteria, which they were almost sterile already from the pupal stage. Now, we wanted to know if our oxotrophic E. coli was able to support conventional larval development. The first thing we tested is the duration of development. You can see here the duration of first instar larvae to L2, L3, L4, pupae, and adults. And we saw that overall the dynamics were really similar. We also looked at the growth of our individuals by measuring larval length 1 week after starting the notobiology colonization, and also in adults quantifying wing length, which are actually readouts of, the wing length is a readout of the size of the adult. And in both cases, in the larval and adult stages, we did not see any difference between our 3 conditions, indicating that yes, Escherichia coli oxotroph for mesodiaminopimelic acid and D-alanine is able to support a conventional development when provided these 2 amino acids, which is required for its maintenance in the host. So then we thought that we had a great tool to now get rid of these carryover effects that I mentioned before, because we were able to remove the microbiota between larval and adult stages and look at really the impact of the microbiota on adults. To do that, we still had again to develop a bit further our tool to be able to keep the sterile mosquitoes sterile even after emergence. To do that, we used some sterile plant rearing cups, and we still had to do a bit more of do-it-yourself with designing some 3D prints that we then put inside our cups to adjust it to the needs of the mosquitoes. We then looked at the reproduction success of our mosquitoes. You can see again here conventionally reared mosquitoes, those which are populated with wild-type bacteria, and here those that became germ-free and had been colonized with auxotroph strains before. What we saw is that between those infected with colonized with wild type or or the germ-free one, we had no difference in either the proportion of egg-laying mosquitoes or the clutch size. Here, each dot represents the clutch size of one individual female. So the number of eggs that she laid. And interestingly, as I told you before, not all the mosquitoes here are in the wild type condition are still carrying bacteria after emergence. But even if we take into account only those mosquitoes which do have bacteria, we still did not have any significant impact. We also looked at longevity, and we saw that the time of the lifespan was not different whether mosquitoes, adult mosquitoes carried wild-type bacteria or if they were germ-free. And this, you can see here, the survival of males and of females. I would like also to point that if we, when we did these experiments, we saw that there was some variations between replicates, and it's just for me a way to also point that it's really important to do independent replicates, because even if we are in very standard conditions, our mosquitoes here, they are in climatic chambers with very neat conditions of temperature and humidity that are very controlled. We don't have any modification in the microbiota composition, and even in this case, we still saw significant differences between replicates. So just insisting on this point, that never get rid of the importance of independent replications. So I just want to put this non-effect that we apparently saw in a wider context, because there has been a study before who studied the importance of the mosquito microbiota on egg productions. And what they did in this case is that they took some control mosquitoes that were conventionally reared and removed the microbiota. And what they saw is that, in fact, there was some impact on egg production in these mosquitoes. And also they looked a bit further at it, and they saw that, in fact, in their microbiota, they had some hemolytic activity. And so they tested if, when giving a pre-lysed blood meal to their mosquitoes, so like 50% of the erythrocytes had already been lysed before being offered to the mosquitoes, then they did not see any impact of the antibiotic treatment on egg productions by the mosquitoes. So it suggests that really, when I said before that we can look in presence, absence, as I just showed you, or looking at different compositions, it is something that is important to take into account here. And maybe if we would have, instead of Escherichia coli, used a strain that is hemolytic, we might have actually seen some difference. And so that's an important point to make, and to put our results in a slightly wider context. And this is still something that we have not looked at. So then another thing we thought our hammer would be interesting for is to also look at the importance of the E. coli on larvae, because we know that at the very beginning of the larval development, bacteria are important, or bacterial colonization are important to allow development. But what would happen if we remove colonization a bit later during development? So we decided to look at this and look at if we would be able to stop colonization during larval development by just transferring our larvae, which are here, in a medium that did not contain any supplement of amino acids anymore. we did this, and here is what you can see on this left-hand chart. If we look at the blue, the auxotrophic colonized bacteria, and this is just the time before transfer, and then we transferred them in a medium without any amino acids. And after the following time points, we looked at the number of bacteria in each larva. We saw that the amount of bacteria was really drastically reduced already 2 hours after transfer, and that they were almost completely lost by 12 hours after the transfer. And interestingly, we saw that there was also a drastic drop in bacteria in larvae that were transferred and did carry wild-type bacteria, and that then this number of bacteria increased again, and by 20 hours, we got back to normal values in number of microbiota. So then what is the impact of this decolonization on development success? Here you can see the proportion of L3 that were able to go to L4 after having been transferred, or go to pupae or to adults. So going to the different next stages. We saw that although all of them in any condition were able to go to the fourth instar of larvae, only 20% of the auxotrophic carrying bacteria which had been transferred would be able to go to pupation, and only 20%, like the same proportion, would then be able to go to adults, suggesting that the bacteria are really important even at later stages of development. And we also saw that there was a drastic reduction in the number of larvae that would get to adulthood, even if they had been transiently lacking bacteria. This is in the case of those that were transferred and did carry the wild-type bacteria. So the light orange here are those that were transferred and then got back to a normal number of bacteria. So even a transient lack of bacteria does affect development success. Now we wanted to address the mechanisms of the importance of bacteria during development. How do bacteria support larval development? 2 models had been proposed, and 2 by very convincing studies. One showing that bacteria use up some oxygen in the mosquito gut and create some hypoxia, and they suggest that this hypoxia functions as a signal for mosquito development for each larval stage to the next one. But another study showed that even in absence of bacteria, using very specific diets and specific conditions, you can be able to produce adult mosquitoes, suggesting that bacteria would more be important for the metabolism of the host rather than by this hypoxic signal. And so to assess these mechanisms, we decided to use our hammer and to do a transcriptomic analysis by RNA sequencing. After having colonized our germ-free larvae with either strain of bacteria, and then having transferred them to non-supplemented medium in the case of the auxotrophic ones, and then 12 hours and 20 hours later, we would sample the guts or whole larvae. Here is the principal component analysis of what we did get after RNA sequencing. We saw that the first principal component discriminated different timings in development, 12 hours or 20 hours after the transfer, while in the guts, the second, like in both cases, the second principal component discriminated wild-type or germ-free conditions. And interestingly, this second axis was already able to discriminate guts from the 12th hour time point, while in whole larvae, you needed to wait for 20 hours before being able to really discriminate those two types of samples. So we saw this observation as a validation of our experimental setup, because in gut, then 12 hours is already a good time point where you can discriminate your samples, while you need to wait 20 hours for the whole larvae. And then we did a KEGG enrichment analysis to know which pathways were involved in this, were particularly up or down regulated. We found that the folate biosynthesis pathway was particularly highly upregulated, and with alkaline phosphatases being the genes that were the most highly regulated of our set. And some pathways were down-regulated and correspond to hydrolysis, transport, and storage of proteins pathways, such as hexamerins, and lipids, such as lipases, lipofluorine, or vitellogenin. So going a bit further onto the folate pathway, what we saw is that it does increase development success. So we wanted to know if it increases, if it is indeed one of the most important pathways involved in mosquito development and linking with the presence or absence of the microbiota. First, looking a bit further on these results that I've just shown you, here you can see the name of several enzymes which are regulated in the mosquito in absence of bacteria in the mosquito larvae. And here in orange, these are 3 genes that are missing in the Aedes aegypti genome and present in the E. coli genome, suggesting further that probably E. coli needs to provide at least some metabolites of the pathway to their host. To further test this, we decided to provide folate to our larvae after having transferred them so that they become germ-free. And so all of these are germ-free larvae, being or not provided with different concentrations of folate, as you can see here. And we saw that the proportion of germ-free larvae reaching adulthood increased after folate supplementation compared to the germ-free larvae that were non-supplemented. And this was not really significantly different depending on the composition in folate. So, but now when I showed these results several times, people asked me, but what about endosymbionts, and is Wolbachia able to rescue development of otherwise germ-free mosquitoes? So Wolbachia is a bacterium that is present in some insects and which is present inside the eggs. So for instance, if you would like to produce germ-free eggs of these species, you would still have Wolbachia being part of it. It's not the case of Aedes aegypti, which doesn't naturally host Wolbachia, but it is the case of other species. And for instance, Culex quinquefasciatus, which is a mosquito present in French Guyana. And so we decided to test if Wolbachia itself was able to rescue development of otherwise germ-free mosquitoes. So we collected these mosquitoes. As you can see, we just have to put a bucket like that on the campus, and we can collect some eggs that have been laid by the wild-type females. This project is ongoing, and it's done by Danai Bemplidaki, who is a master's student in the lab. So they are Culex eggs which carry Wolbachia. Then we do sterilization, similar to what I told you before, and we can get some germ-free larvae. And then what happens if we add E. coli or if we don't add E. coli? We saw that in the absence of E. coli, even after waiting several days, larvae would just stay at the first instar, would maybe grow very slightly, but almost nothing. And also a lot of them would even die. While in the presence of E. coli, a lot of the larvae would develop to adults and in kind of standard conditions. We also had to validate that our larvae did carry Wolbachia. This has been fully validated with bigger larvae with L3 or L4 after having been colonized with Escherichia coli. And with those that were fully germ-free, they are smaller, and so far the results are a bit less clear, but it looks like we indeed have Wolbachia. This is very much work in progress. So, but as I told you before, you can work on the presence of the microbiota, but it's also interesting to look at its composition. And here is the cycle of the mosquito, where the female would lay eggs, then forced instars of larvae, pupae, and adults. And if you look at the cycle of its microbiota, there is a bit of the sources of the microbiota. One thing is that each stage would transmit some microbial partners to the next ones. And for instance, females would deposit some microbes on the egg surface, and also in the case of endosymbionts, inside the cytoplasm of the egg. And then from each stage to the other, there will be some transmission. And but the environment is a main source of the microbes. For instance, in the adult, the adult would ingest some breeding water when it emerges, and thus be colonized with microbes from the breeding water. Then it can be colonized by microbes coming from the environment in general, and notably from the diet or the mating partner. The larvae can be infected or colonized by microbes coming from the breeding water, and part of them also from other organisms through the feces or the dead bodies of these organisms, for instance. And also at the end of the larval development, just before becoming pupae, larvae discharge most of their microbes in the environment, which then can be ingested by the younger larvae. But as environments are diverse, they also have diverse microbial compositions, and also between laboratory conditions and field conditions, you will have very different environments and also thus different microbiota compositions. And so the main question we had when starting this project is, can we produce mosquitoes with a relevant microbiota composition? And thinking about this question led us to think about the hypothesis of the presence of enterotypes in the human gut microbiome. This has been suggested by Arumugam and co-authors in 2011 and has been intensely debated since then. The idea is that because some bacteria would have positive or negative interactions between each other, you may have some compositions which would be possible in the human gut, while others would not be possible. And that if you look at the composition, then you would be able to kind of type people depending on the composition of their microbiota, because some microbiota compositions are more frequently found than what would be random. And so actually what they did is that they sequenced the whole genome of the microbiome and found 3 different enterotypes. And just taking this as a hypothesis, if it exists in mosquitoes, it would be interesting maybe to then try to graft them in a simplified version in our germ-free mosquitoes and see what is the impact of microbiota composition on transmission. So what is known so far is that there are positive or negative correlations between the presence of different strains of bacteria in mosquitoes. This has been found by Hegde et al. In 2018. And now, can we graft simplified enterotypes to our germ-free mosquitoes? What are 2 arguments that are interesting is that one is that there is a low diversity in the mosquito with 3 to 4 bacterial species or bacterial OTUs, more specifically operational taxonomic units. That's what we call what we get when we do sequencing of the sequencing studies. And these 3 to 4 bacterial species represent 80 to 90% of the reads in one individual mosquitoes. Moreover, many genera which have been found in culture-dependent methods and culture-independent methods on the mosquito microbiota composition are similar, and so suggesting that many genera of bacteria colonizing the mosquito are actually culturable in standard in lab conditions. And this can be also due to the fact that there is no strict anaerobic condition in the mosquito gut, but that it's more like normal normoxic or hypoxic conditions. And so if we can graft these simplified enterotypes to our mosquitoes, we would be interesting to see if there are some transmission enhancing enterotypes or transmission blocking enterotypes, and then which environmental factors would shape each of these enterotypes and thus have some positive or negative impact on transmission. I will not answer on any of these questions that I just raised. It's very much work in progress, but just to give you a bit our way of thinking about the question of production of relevant mosquitoes to study the mosquito microbiota interactions. I just still have the hypothesis of the presence of the mosquitoes in the enterotypes in mosquitoes. This is very preliminary data, which we are still looking at if it is validated or not. But to tell you how we do that, we go to different places in French Guiana, go with our traps called mosquito magnet, and we collect mosquitoes and then do the analysis of the microbiota composition in single individual mosquitoes and only in the guts of these mosquitoes. So it's really tiny samples, and that's why we are not completely sure, and we are still doing some validations of our protocols. Here, if it is the case, we have identified 2 different types of microbiota compositions which were likely to happen in our 2 conditions. One that corresponds to all the samples that were collected in Trois-Palais-Tuviers. It's a riverside village in the village on the Oyapock River, which borders Brazil, and the other one is collection of La Césarée, like a rural area more present in a savanna landscape. But as I said, very much something that needs a lot of validation before we can really say that we found any enterotypes in mosquito. But while doing these collections, we had to develop some tools to do taxonomy for beginners. Indeed, we are not experts in taxonomy, and when we are collecting mosquitoes, we have around 5 to 10 different species of mosquitoes which are present in our collections. And when you want to identify the mosquito species, there are different types of situations. Either you have a very high level of diversity, and in this case, what you would do is that you would extract the DNA, do a PCR, and sequence, like PCR, for instance, on the ITS2 gene, that is a gene often found for analysis of the mosquito species, and then you sequence it, and it will tell you which are the sequences from. If you deal with very low level of diversity, then you can either train everybody to do a morphological analysis, or design some primers so that you can do multiplex PCR and that you have bands that are very with very different sizes, and so that you can identify which mosquito you are dealing with. But in our case, we were with an intermediate level of diversity with 5 to 10 different species that we wanted to discriminate. And as you can see here, they have, this is the size of the bands of the ITS2 amplicon that we find in 5 of these species. So what we decided to do is to use also a method based on DNA extraction and PCR on ITS2, but then we run it on a capillary electrophoresis system called Kaya-XL, which is able to discriminate between around 3 base pair precision the size of the amplicon. And we run it in our system. And you can see here the results we would get with the different size that we got with something of the different species. And you can see that most of them, we can discriminate them according to the size, but we still needed a little more information to be able to discriminate some species between each other. And for this, luckily, we also were able to look at the tip of the leg of our mosquitoes. So even not being a very expert in taxonomy, this is something we would be able to do. So most of them, so all of these species have some white region at the end of the hind leg, and some of them have a little black dot, or some are white until the end of the hind leg. And by discriminating just via the Kaya-XL analysis and with adding this little morphological analysis, we were able to identify 91% of our samples via the Kaya-XL, via this method, and with 9% of our samples being still uncertain. So we think that with these 9% of uncertainty, you could still either send these samples for sequencing if you really want to address a sample, to use all your samples, or just consider that these samples would not be a candidate for further analysis, whatever you want to test. And another thing that, in terms of tool that we had to develop further when doing these microbiota analysis is dealing with the problem of contamination when sequencing very low biomass sample. As I told you, we are working on mosquitoes, but then we are using the guts of mosquitoes, which are even lower source of in terms of amount of bacteria. And even among these mosquitoes, among these mosquito guts, when you look at microbiota from field mosquitoes, they even have less bacteria per mosquito than what we would typically get on lab mosquitoes. So it's really a very, very, very low biomass sample. So it is known that there are problems of contamination in these type of samples. So what you can do, the first thing is to try to avoid contamination. So work under a microbiology safety cabinet, aliquot kits, use kits that are very specific to microbial composition, but then with very low biomass sample, you cannot get perfect data. And so looking at different publications, there are 3 different types of approaches that were used to deal with the negative controls. So negative controls are some samples which can contain nothing, and where you would just keep together with your other samples throughout extraction or PCR, or at different stages you can do some negative controls, and then you sequence them and see what is present in them. Some publications chose to just remove all the bacteria which were present in negative controls. Some others would just report them as contaminant and check that they were different. They found some differences between their controls or not, and from them samples. And the others would identify what I would say real contaminants, like they put some types of threshold to identify contaminants and then remove these ones specifically. But in general, all these publications would never show much information on negative controls. And what we suggested by a recent project that we did in collaboration with our partners in Burkina Faso, and specifically Aminata Fofana, who is a PhD student, we suggest from our data on single gut analysis to first identify some major contaminants, which we think need to be highly prevalent, and also to have a relative contribution higher in samples, higher contribution in negative controls than in samples. This point is very important because if you remove all the contaminants which are present in the negative controls, you may remove some which are actually cross-contaminants present in the negative controls because they came from the samples. And so these ones, you really don't want to remove them, while you would like to remove those that were highly present in the negative controls and probably show that they contaminated the samples thus. And the second point we suggest is that it is important to report negative control data throughout the study. And to highlight this, I will give you this example where you can see three types of samples, and here some negative control data. This is the alpha diversity, which is supposed to refer to the diversity present within a sample. And if you don't see any negative control, you would have the impression that this type of sample here on the left-hand side would have a higher diversity. But if you do see also what is found in negative controls, then you would think maybe in fact you had such a small number of bacteria here that actually each little contaminant has been detected, and that actually this is the reason why you may actually see higher diversity, and this is very similar to what you get in negative controls. So maybe in fact what is the result is that you would have less bacteria in total. So at least giving these negative control data would really influence the conclusions you would be able to draw from your results. And that's why we think that it would be interesting that everybody reports the negative control data throughout studies when dealing with such low biomass samples. Going further on our tools, when we want to repopulate our mosquitoes with different types of bacteria, we also decided to build a library of bacteria. So to do that, we collected mosquitoes, isolated different bacteria, and did a biobank where we are identifying species, and then also looking at the characterization of these bacteria, where they have proteolytic or hemolytic activities, and also do a bit of in vivo characterization, looking at the effect of these bacteria on mosquito fitness, vector competence maybe in the future, and also the physiology a bit further. And this is an example that you can do. So this with our Serratia marcescens strain that we isolated from actually lab mosquitoes, we did some analysis either of oral infection in adults, where we saw quite low virulence with only some virulence after 10 days of infection, and it's not very major virulence. While when you use the same quantity in larvae, I'm just focusing here on the red part, which is the wild type bacterium. We saw a high virulence with like 80 to 90% of the mosquitoes dying during larval development. And this was also still found, even if we would further dilute to 1,000 the number of the bacterial suspension, suggesting that virulence in larvae and adult would be different. And this is just to give an example of what we can do with bacteria of our colony. And the final tool I wanted to introduce you is about outreach. So we designed a set of tools to be able to work in schools with some students from different ages, from memory, trivial pursuit, or puzzles on mosquitoes. Here you can see that some of the kids are really small, like from 4 years old or something like that. And sometimes we also went to secondary school to build a scientific project where students were involved from experiments to the analysis of their results and even to poster construction. And some of them even used it in an oral exam that children around 14 years old have to do in France, and it's called the Brevet des Collèges. And it was a very important tool for them to be part of, to participate, to propose this as their main project of this oral final exam of this intermediate secondary school. It was really a success, and we are doing it again. And I just want to put it a bit in a context. We decided to use to propose education tools as a benefit sharing for populations in French Guiana, because we are collecting mosquitoes, which besides nuisance or vectors are also genetic resources. And so to conform to the Nagoya Protocol, we had to give some benefit sharing to the society. And that's what we used. But in fact, it was really rewarding, and I really encourage anybody to do such project. It's rewarding for kids and for us as well. So in conclusion, we have a toolbox that allows us to produce germ-free mosquitoes at any stage, at least in Aedes aegypti. We are still working on other species to continue. Taxonomy for beginners, sequencing analysis from low biomass samples. We have a library of bacteria and outreach tools. Some of the tools are present on the do-it-yourself section of our website, and some will be available in the future, and don't hesitate to contact me also if you need some of them. To conclude, I would like to thank everyone who was involved in all this work, notably all the members of the team. Here is more or less the first generation and second generation of the Microbiota of Insect Vectors group, our collaborators, and our funders. In particular, the ANR, which is our main funder, partly via the LabEx IBID. And I thank you for your attention. I would be glad to take any question.
Okay, Mathilde, nice presentation. Just a moment. Je vais dérouiller mon français aussi. One more try me. Je remercie par avance Mathilde pour essa apresentação que apportei plusieurs aspectos da importância da microbiota dans le développement de moustiques ou différentes étapes de leur cycle de vie. I was trying to write this before you appointed your last topics about the microbiota and the taxonomy. I was particularly interested in this topic also, and the work with the children. So I did not point at these aspects in my abstract of your presentation. The audience is very shy, so I have some questions to begin our discussion. The first one, Mathilde, is about viruses. I know that you are addressing just with bacteria composition, but what about viruses? It's possible to sterilize eggs from transovarial transmitted viruses, because I note in your protocol of sterilization of eggs in the chapter you recently published with Otavia, and I was trying to apply with Sandfly, but this is my second question. The first one is about viruses in the interior of eggs and the possibility of sterilizing eggs with this technique.
I don't think that if there would be any virus in our mosquitoes, no, I don't think we would be able to sterilize them with this technique, because all the DNA that is present inside the egg would not be affected, and so then also the internal viruses inside the cytoplasm of the egg would also not be cleared. So what we can see is that anyway, whether they are or not cleared, then what we looked at is we started always with the same batch of eggs that we then populated either with E. coli wild type or oxotrophic E. coli. And so whatever the presence or absence of these viruses, then it would not, this is not something that we are able to look at, but also it would not change our setup because, it's part of what is present inside the egg from the beginning. So unless it really has some impact on the bacteria itself, then we think that it's not a huge problem. But yeah.
Okay. It's a challenge for us because we need to sterilize from transovarial transmission of some virus. So the second question was about
and
sandflies.
also I just because I just to say we did some transcriptomic analysis and we didn't find any viral DNA, but in fact the way we did with our transcriptomic analysis, we used some poly-A tail selection of the mRNAs specifically. So for instance, I could not say that we didn't detect any viral DNA via this way because actually if there would be any, we would have lost them before. So it's not, I'm really not able to say whether there are some or not.
Okay. The second question, I need to talk about sandflies because it's my expertise. So what about sandflies? How easy or difficult do you think the toolbox you presented now it's applicable to these insects? My question is driving specifically because the life cycle of sandflies is not aquatic, it's terrestrial. So some methods of sterilization of different stages of life cycle of mosquitoes need to be immersed in water. So how do we can do this method with sandflies? Could you help us?
I don't know, but what I know is that so the sterilization doesn't last very long, and it's like 5 minutes, like in each washes, and then you wash it in water. And for instance, that is very similar to what you would do with Drosophila eggs, where you also would sterilize them, I think, with bleach technically before you, and then rinse, and that's how you get some sterile Drosophila eggs. And also they are not. They don't develop in water. They also develop on some type of solid medium. So I don't think that the fact that it is not developing in water would be a problem. But I've never dealt with any sandfly colony, so I don't know much more than what. Yeah.
Yes, you wrote also that you use 15% more eggs, for example, because you have you lost some eggs during this sterilization. How much percent is the lose?
I don't know, but
sterilization? Because
really,
you need to hatch the larva to account the viability of the egg in this possible impact of sterilization into the viability of the egg.
So I don't know exactly how many eggs after sterilization go to the first larval instar, but what I know is that after what we do is generally we sterilize the eggs and then put them in a cup, and then we take only the L1. So then we didn't really quantify the success to hatching to the first instar of larvae. But what we found is that from the first instar, definitely you had really no problem in development until adulthood. So, and we didn't have the impression that many eggs would die during this process. But yeah, we didn't really count specifically the proportion.
Okay.
Does it answer the question? Right?
Yes. It was clear. Another one, the microbiota. Did you wrote this statement in your recent paper? I think that was with Biotava also about the microbiota, non-intestinal microbiota composition. particularly with the salivary glands and the reproductive tract. So, do you think that this microbiota specific impact on the transmission and also with the fitness of the mosquito, not just with the composition of the intestinal microbiota, but the reproductive tract and the salivary glands, because we have a specific composition of microbiota, and how much easy or difficult is to study composition, different composition, microbiota composition in the different parts of the mosquito due to the small size. So how much easy is to isolate bacteria and be sure that they are compounded from the salivary glands and not from the intestine, for example. So some questions about this subject.
Yeah, so some people have dissected specifically salivary glands and guts and then genital tracts. That's the main places where people are interested in looking at the mosquito microbiota. And indeed, there are some differences can be found, but in fact, what has been found by the lab of Isabelle Morlet is that there were more differences between individuals and depending on their origin, for instance, the larval breeding site or something like that, rather than or different mosquitoes coming from different conditions, whether they were fed differently or something like that, sugar fed, blood fed, or something like that, compared to the difference between individuals. And this is it's quite different to what you would see in humans, for instance, where the microbiota of a gut would be more similar to microbiota of another gut, and than the microbiota of the gut compared to the vagina microbiota, which is more different than going from individual to individual. So it's like, it looks like tissue is not the main driver of the difference and of the microbiota in like in mosquitoes. It's also interesting to think more functionally, as you said, like, is the microbiota from the salivary glands particularly interesting or having some impact? And I don't know how you could definitely make a different microbiota, whether in presence, absence, or composition in the gut and in the salivary glands to discriminate the impact of each other. But yeah,
very challenging.
You would have to develop more tools to do that. And also, what is present in the saliva may also be ingested inside the mosquito and have some impact inside the mosquito gut. So, yeah.
Okay. J'ai besoin de parler d'argent, cost effectiveness, because you talked about the applicability of these studies into the mosquito control, so the surveillance of disease-based agents. So what about the cost effectiveness of methods of manipulation of insect microbiota? How much does it cost for research? And it's really in the future applicable for mosquito control, including the topic of endosymbionts and Wolbachia that you addressed in your presentation also, because it's very, so in Brazil, you talked so much about Wolbachia into this subject of future control of disease, especially with dengue and other arbovirus transmitted by Aedes aegypti. So what about cost effectiveness of these methods?
I'm not the best person to say that, but you know, like the use of Wolbachia has been started in several countries now, and now it's a question of where you want to put your money. And so I think it's Malaysia now who decided to invest money that used to be invested for conventional insecticides now into Wolbachia control. And so then it looks like if they decided so, they probably were convinced that it is cost effective, and that and indeed they had some interesting impacts on reducing dengue cases in their country. And like now there is this World Malaria Program which has started in one country and is now present in 12 or I don't know how many different countries. You would not convince a lot, like many countries, if you would not be cost effective. And so I don't know much about the cost effectiveness myself, but what I can see is that it has already convinced some people who are probably much more expert about that than me.
Okay, no problem. Just a question of my mind. I need to talk also and again with the virus because you addressed a question of preliminary work about the enterotypes in mosquitoes, because you are trying to make some field work in Trois Palais with. So some insect-specific viruses are a basic composition of the gut of some mosquitoes. It's possible to link the search of microbiota composition together with the virus composition to fill the catch flies. And if it's important to link these two kinds of research.
Yeah, I mean, it would be really great if besides our analysis, we would also know what are all the different microbes. And I think that looking at, for instance, fungi may probably not be very different.
Fungi also, sorry.
Yeah, but looking at viruses then would add, in terms of cost of sequencing, would probably add much more. And so that's more the reason why because we were speaking about cost effectiveness.
Yes.
I mean, that's probably the reason why we decided, in fact, to look at many different samples, to look at our enterotypes, rather than having a smaller sample set that we could really analyze in big details. Maybe, I don't know, maybe one possibility is that then when we have this defined, whether we have enterotypes or not, we could, on a smaller set of samples, do a really whole like really sequencing analysis without any amplicons, but analyzes all the nucleic acid of the samples.
Okay. Do you test it's the other topic? Sorry, change the channel. Do you test the microbiota manipulation of microbiota into different stages of the life cycle of lab mosquitoes? I'm not sure that I understood into one generation. My question is driving to that I make the keyword aqui. Environmental and microbiology pressure in lab conditions impact in long term of generations. For example, 10 generations after under microbiota manipulation, it could be impact in the fitness, measure and life cycle. Not just one generation of observation, but a long term of observations. If there's some impact into the genetics of the mosquitoes in long term, and consequently with the phenotypical characters of mosquitoes in long term, 10 generations or more of observation. It's possible to drive this question, this hypothesis.
No, but we didn't do it ourselves, and I know that some people have looked at like 3 generations of mosquitoes in the lab of Jacobs Lorena, for instance. I don't know much about if people have gone to like 10 or 20 generations, but definitely I know that there is interest in checking these type of questions because of it's important to test potential if you want to then go to implementation of the use of some microbes in the field, then of course you need to know if they are able to infect mosquitoes, colonize them for many generations, and then also not affect too much the fitness of these mosquitoes for this number of generations, so that these microbes can also be kept in mosquito populations. But yeah, it's not really. I'm not. I don't know much more about that.
Yes, I was addressing this question because you wrote that use linear morphometrics to measure the size of wings and the whole larvae, né? And use also geometric morphometrics to address some questions about the genetic difference between some populations. So I think that in long term of environmental pressures of microbiology, microbiological driving pressures, we can note in a fine scale some difference of geometry, especially with the wings, because it's the main structure to be studied between mosquitoes, and to hypothesize different populations driven by some events like environmental pressure. So that's my question is driving, but specifically with about your methods of measuring of fitness in the mosquitoes, like morphometrics.
Yeah, it's true that I said, and that's why also I didn't want to say too much that the microbiota doesn't have an impact on adults, because I just said that with our conditions with E. coli, we did not detect any impact on significant impact on the fitness parameters that we tested. And yeah, it's true that if we would have gone to a longer term experiments, maybe we would also have seen some impact. I don't know, but at least in the conditions we have seen, we were not able to detect any difference.
In your last, sorry. In your last point, your last was about the Brevet de Collège. The other one about taxonomy. So in French Guyana, there was writing some works about Mauditoff or protein-based methods for taxonomy. In your opinion, DNA sequencing or Mauditoff or morphology to improve the taxonomy of mosquitoes in French Guyana for now? Because you defended about sequencing for now.
No, I think that Mauditoff is also a very interesting tool if you have it locally. I mean, it is really interesting as well. Yeah, I mean, the thing that we had is that for us it was really convenient to do the DNA extraction that anyway we would also need for other purpose, and to use it for, we are kind of not destroying our sample, and we can also redo the analysis whenever we want. And also we don't have a Mauditoff on site, so it's more easy to do this type of analysis. I think that there is not one or the other, but that several tools sometimes may be interesting.
Okay, very nice. So, Mathilde, no more questions. The audience is very shy, but I think this meeting can be shared after this presentation. That's because I warranted to you that do not share unpublished results. So I would like to thank you again and the team of Institut Pasteur that you take part to share these interesting results, and talk also that we transmitted this live now from Belém, Brazil, via the PPG BAIP YouTube channel. Thus, these transmissions can be accessed permanently from anywhere in the world. So we talked about the tripartite interactions between the mosquito and its microbiota, with Mathilde GENDRIN. She's a specialist in this subject and work not so far from us, because French Guiana is just 1 hour and a half from fly. And I need to ask about possibilities of collaborative work, because we have an audience can be very interested in work with microbiota and mosquitoes. So you can talk about this possibility, if it is possible, with Institut Pasteur and Universidade Federal do Pará. It's possible
of course,
to.
people can contact me when they want to discuss anything.
Okay, so I would like to acknowledge to everybody, to this audience, and thank you for your presentation, your nice presentation, Mathilde. Hope to see you in another moment in the world, here in Belém or in French Guiana or in any place in the world that it could be needed to meet about this subject. So my acknowledgement is special to you.
Or about sandflies as well.
Yes, about sandflies. Yeah. It's my main question about this expertise.
Thank you very much.
Okay, good morning.
Bye.